Paukner. I'm presenting Lefamulin for Nabriva Therapeutics. Lefamulin is an approved antibacterial, approved for the treatment of community-acquired pneumonia in adults. And we investigated the activity of Lefamulin against Staph aureus collected from cystic fibrosis patients who were hospitalized with pneumonia. So Staph aureus is the most often isolated organism in cystic fibrosis patients in the young patient population. Although there are some treatment options available, orally and IV administered, there is still a high medical need for treatment, for other treatment options, because all of the available treatment options have shortcomings. Either is it the lack of evidence-based treatment guidance or side effects. So what we did is to collect Staph aureus isolates during the past 5 years of our surveillance study globally from many centers, medical centers in the world. We included 269 bacterial isolates in that case and tested against Lefamulin in comparators according to CLSI methods. In table 1, you will see the MIC testing results showing Lefamulin and the comparators. And you will notice that Lefamulin was highly active against the collected Staph aureus isolates with MICs MIC 50, MIC 90 of 0.6 and 0.12 microgram per ml. And we saw 100% susceptibility, taking a breakpoint of 0.25 microgram per ml into account. So this is the breakpoint approved by the CLSI, FDA, and UCAST. Specifically against the MRSA subpopulation, so we had 55 isolates included in that population. Lefamulin displayed potent activity, also with an MIC 90 of 0.12. It was highly active. And you will also notice that comparators showed really high resistance rates. For example, azithromycin, here more than 73% of the collected MRSA isolates were resistant. Also, the levofloxacin resistance rate was really high, with 36.8%. So this illustrates that it's really that new treatment options are required. This figure also shows you the MIC distribution. In green, Lefamulin, showing, illustrating the MIC distribution and the maximum MIC of 0.25 microgram per ml, while all the other comparators displayed higher MIC values and also resistant subpopulations. So to conclude, in that study, Lefamulin demonstrated potent in vitro activity, regardless from the resistance phenotype. And these results, in addition to other favorable properties demonstrated in earlier studies, suggest that Lefamulin may be a viable treatment option for cystic fibrosis patients with exacerbation due to Staph aureus. The other results that were not shown on that poster are that a similar PK and safety profile is seen in cystic fibrosis patients as in healthy people, and the drug-drug interactions with CFTR modulators, ivacaftor, is very low. So this was demonstrated in a clinical trial recently published. And finally, Lefamulin also displays anti-inflammatory immunomodulatory properties. So taken together, these data suggest that Lefamulin may be a good treatment option for cystic fibrosis patients with pneumonia due to Staph aureus. So finally, I would like to thank the entire NABRIVA and JMI teams for supporting this work presented on that poster.